Here's something that should terrify every biotech researcher on the planet. We just watched an AI solve a 15-year-old puzzle in a matter of weeks. And if you think this is just another tech hype story, stick around. Because what OpenAI and Retro Biosciences pulled off in early 2025 might be the biggest leap forward in reversing human aging we've seen this decade. Picture this. Inside your body right now are adult cells that have a specific job. Skin cells, liver cells, heart cells. Back in 2012, a scientist named Shinya Yamanaka discovered something incredible. He found 4 special proteins that could essentially rewind these adult cells back to a stem cell state, like hitting the undo button on aging. It's the closest thing we have to cellular time travel, and it won him the Nobel Prize. These proteins, called the Yamanaka factors, have been used to treat blindness, reverse diabetes, address organ shortages, and combat infertility. They're like the master key that unlocks your cell's ability to become young again. But here's the brutal reality nobody talks about. They barely work. Imagine you're trying to reprogram 1,000 old phones back to factory settings, but only one of them actually reboots. That's the success rate we're dealing with. Less than 0.1%. It's like trying to fill a swimming pool with an eyedropper, one drop at a time. For 15 years, the best minds in regenerative medicine have been trying to improve these proteins. Traditional methods involve changing 1 or 2 amino acids at a time, testing thousands of variants, hoping to stumble on something better. 15 years of chimeric SOX protein research yielded variants that differ from natural versions by only 5 amino acids. 5 out of hundreds. Now, let me tell you what just happened at Retro Biosciences, a longevity biotech startup that partnered with OpenAI's research team. They created something called GPT-4b Micro, a specialized miniature version of GPT-4o trained specifically for protein engineering. And when they turned this AI loose on the Yamanaka factors, it didn't just make incremental improvements. It redesigned these proteins from the ground up, changing more than 100 amino acids on average, and achieved something that sounds impossible: a 50-fold increase in stem cell reprogramming markers compared to the original versions. 50 times better. Let that sink in for a moment. Before I break down exactly how they did this and what it means for your health span, we need to talk about why this approach is so radically different from anything that's come before. Because the really wild part isn't just that AI made better proteins, it's how it learned to think about proteins in the first place. Most protein language models are trained exclusively on protein sequences, like teaching someone to write novels by only showing them individual words with no context. But GPT-4b Micro was different. OpenAI initialized it from a scaled-down version of GPT-4o, which meant it already had a broad base of biological knowledge and language understanding. Then they trained it on protein sequences, biological text, and tokenized 3D structure data, elements that most protein models completely ignore. Here's where it gets fascinating. They enriched a large portion of the training data with contextual information, textual descriptions of what proteins actually do, co-evolutionary sequences from related proteins across different species, and groups of proteins known to interact with each other. This context allowed the AI to be prompted with specific desired properties and generate sequences accordingly. It's the difference between asking someone to write a compelling story versus just asking them to string together grammatically correct sentences. And because most of the data was structure-free, the model handles proteins with intrinsically disordered regions just as well as structured proteins. This is crucial because the Yamanaka factors aren't rigid, neatly folded molecules. They're proteins with long, flexible arms that need to form hundreds of transient interactions with diverse binding partners. Traditional protein design tools struggle with this kind of target. GPT-4b Micro excels at it. But in silico evaluations, computer benchmarks, they only tell you so much. The real test is whether this thing works in actual human cells. So, Retro's scientists built a wet lab screening platform using human fibroblasts, skin and connective tissue cells, and asked GPT-4b Micro to propose a diverse set of enhanced SOX2 variants, which they called retro-SOX. The results were staggering. Over 30% of the model's suggestions outperformed the original SOX2 at expressing key pluripotency markers. Remember, these variants differed by more than 100 amino acids on average. Traditional directed evolution screens typically see hit rates below 10% when changing just a handful of residues. Getting a 30% hit rate while making massive sequence changes shouldn't be possible. But it happened. Encouraged, the team tackled KLF4, the largest of the Yamanaka factors. Prior attempts to improve KLF4 through expert-guided single amino acid substitutions had produced one hit out of 19 tested variants. GPT-4b micro-generated retro KLF variants with a hit rate of nearly 50%. 14 model-generated variants were superior to the best cocktails from the retro SOX screen. Now, here's where things get really interesting. When they combined the top retro SOX and retro KLF variants together, they saw dramatic increases in both early and late-stage pluripotency markers. Late markers appeared several days sooner than with the original Yamanaka factors. By day 10, cells reprogrammed with the engineered variants showed robust alkaline phosphatase activity. The purple colonies you see in stem cell labs that indicate successful reprogramming. The intensity and number of these colonies were far greater than anything seen with the wild-type factors. But wait, there's more. And this is the part that should make every longevity researcher sit up and pay attention. They tested these retrofactors using mRNA delivery instead of viral vectors, and they used mesenchymal stromal cells from 3 middle-aged human donors over 50 years old. Within just 7 days, more than 30% of the cells began expressing key pluripotency markers. By day 12, numerous colonies appeared with typical iPSC morphology. Over 85% of these cells activated endogenous expression of critical stem cell markers. They then verified that these retrofactor-derived iPSCs could successfully differentiate into all 3 primary germ layers: endoderm, ectoderm, and mesoderm. They expanded multiple monoclonal iPSC lines over several passages, confirmed healthy karyotypes, and genomic stability suitable for cell therapies. Every benchmark consistently surpassed results from conventional iPSC lines generated by contract research organizations using standard factors. This isn't just a laboratory curiosity. This is proof that AI-guided protein design can work across different delivery methods, different cell types, different donor ages. It's robust, replicable, and scalable. Now, let me show you something that ties directly to why you're watching a channel about longevity. The Retro team decided to test whether these re-engineered variants had enhanced rejuvenation potential, specifically their ability to repair DNA damage, which is one of the canonical hallmarks of aging. Previous work has shown that Yamanaka factors can erase DNA damage-related aging markers in mouse cells without fully reverting cell identity. So can the retro factors do better? They treated human fibroblasts with doxorubicin, a chemotherapy drug that induces double-strand DNA breaks, absolute havoc at the cellular level. Then they reprogrammed these damaged cells with either a fluorescent control, wild-type Yamanaka factors, or the retro SOX and retro KLF cocktail. They measured DNA damage using gamma H2AX immunostaining, where lower fluorescence intensity means fewer breaks and better repair. Cells expressing the retro variants showed a marked drop in gamma H2AX signal relative to both controls. The differences were statistically significant. This suggests that the retro SOX and retro KLF cocktail reduces DNA damage more effectively than the original Yamanaka factors. By ameliorating one of the core hallmarks of cellular aging, these engineered variants offer a potential path toward improved cell rejuvenation and use in future therapies. Think about what this means. We're not just talking about making stem cells more efficiently for research purposes. We're talking about proteins that could potentially be used to rejuvenate aged cells in living organisms, reducing DNA damage, restoring youthful cellular function, all the things that break down as we age. Remember that longevity isn't just about waiting for miracle therapies. The fundamentals still matter. Exercise, nutrition, sleep, stress management, metabolic health, these are the foundations that keep your cells resilient while the cutting-edge stuff moves through the pipeline. Think of it this way. Every year you stay healthy is another year closer to breakthrough therapies becoming available. Time is the most valuable asset in longevity, and you buy time by taking care of the basics today. What we're witnessing with GPT-4b Micro and the retro factors is a glimpse of how fast things can move when the right tools meet the right problems. This is what exponential progress looks like in biology. And if you're paying attention, if you're staying informed, if you're taking care of your health with the knowledge we already have, you're positioning yourself to benefit from everything that's coming next. Thank you for spending your time here. I'm grateful you're on this journey with me, wishing you practical progress and good health. If you found this helpful, I've got more eye-opening research on longevity compounds that most people have never heard of. Click the video that just appeared on your screen to discover the next piece of the healthy aging puzzle.